Environmental DNA as a Molecular Tool for Blue Whale Conservation by Shaza, Azim, Arif Mujahid and Radin from AS2446A.
Hi everyone, my name is Shaza Batrisha binti Norafis and I will be introducing the blue whale, which is scientifically known as Balenoptera musculus. The blue whale is the largest animal to ever exist on Earth, reaching up to 30 meters in length and weight more than 150 tons. It is a marine mammal belonging to the baleen whale group that can be found in all major oceans, although it is rarely seen because it spends most of its life underwater. Blue whales play an important role in maintaining ocean health. They help recycle nutrients in the marine ecosystem, which support the growth of plankton and the balance of the food web. Because of this, they are considered an indicator species for ocean health. Blue whales are currently classified as endangered by the IUCN. Their population dropped drastically due to commercial whaling in the past, and although hunting is now banned, recovery is still slow. Today, blue whale face several threats. One of the biggest is ship strikes, especially in busy shipping routes. They can also become entangled in fishing gear. In addition, underwater noise pollution disrupts their communication, and climate change affects krill population, which is their main food source. Because blue whales are highly migratory and spend most of their time underwater, it is very difficult to monitor them using traditional methods. This is why modern molecular techniques such as environmental DNA or eDNA are very useful for their conservation. Next, I will pass to Muhammad Azim.
Hi everyone, I am Muhammad Azim. As Shaza highlighted, blue whales face several threats, yet studying them directly incredibly challenging, because they travel massive distance and spend their vast majority of their lives under deep water. Traditionally, visual tracking methods fall often short. This is where molecular biology completely changed the game with environment DNA, or we call it eDNA. So, what exactly is eDNA? Simply put, environmental DNA is a genetic material that an organism sheds naturally into its surrounding environment. Just like humans, they fingerprint behind. A blue whale constantly leaves behind biological signature as it swims. These traces come in the form of soft skin cells, mucus, urine, and fecal particles drifting in the seawater. By simply capturing a cup of the ocean water, we can capture the actual genetic code of the giant swimming through it. Why this is so crucial for marine conservation? For a species as elusive and endangered as a blue whale, eDNA offers a massive advantage. It is entirely non-invasive. We no longer need to stress the animal by chasing them down for tissue biopsy or relying purely on lucky visual sighting. With eDNA, the ocean itself becomes a biological archive, allowing us to detect their presence dynamically. Up next, Arif will talk to you through exactly how we extract and analyze this hidden genetic footprint from a simple water sample. Thank you.
Hey, I didn't see you there. My name is Muhammad Arif bin Sarun. I'm from AS2446A, and today I'm going to talk to you about how eDNA samples are collected for blue whale conservation. Are you ready? Let's go, baby! Blue whales! Blue whales! Blue whales! Where do we find these whales? Firstly, researchers use visual surveys, acoustic monitoring. What is acoustic monitoring? Acoustic monitoring is basically listening to whale calls and, other than that, known migration routes to identify where to sample these seawater samples. And then, after the whale is located, the water is collected near the location where the whale recently surfaced, dived, or exhaled. The seawater samples are stored in sterile bottles to avoid contamination. Now, we are going to the filtration process. The seawater is passed through a fine membrane filter to filter out skin cells, mucus, fecal particles, and other biological materials. Then we're going to extract the DNA from the materials captured from the filter in the laboratory. Now we're going to amplify and sequence the DNA from the blue whale. The researchers use PCR and also DNA sequencing to identify the whale DNA and distinguish it from other marine biology species. Now, would you look at that, man? Blue whales are cute. I love blue whales. Do you love blue whales? I love them. I love them, baby. Alright, so that's all from me. We're gonna move to the next part. I'll see you again. Bye-bye.
Hi, my name is Muhammad Rahman and my student number is 2023217116. Now I will discuss the application of environmental DNA or eDNA in blue whale conservation. One of the important application of eDNA is detecting the presence of blue whales, since blue whales spend most of their time underwater and travel over vast ocean area. They can be difficult to monitor using traditional methods such as visual survey. By collecting seawater sample and analyzing the DNA left behind the whale, scientists can confirm whether blue whales are present in particular area without directly observing them. Another important application is monitoring migration routes. Blue whale migrate long distance between feeding and breeding ground throughout the year. eDNA allows researchers to track the movement by detecting whale DNA in different locations. This helps scientists better understand migration pattern and identify important habitats used during this journey. In addition, eDNA can be used to locate feeding areas. Blue whale often gather in regions with abundant food sources, such as krill. Detecting blue whale DNA in seawater can help researchers identify this feeding ground and determine which areas are critical for their survival. Finally, the information obtained from eDNA study can support marine protected area planning by identifying important habitats migration routes and feeding ground. Conservation agency can make better decisions about which areas should be protected. This help reduce threats such as ship strike, noise pollution, and habitat disturbance. In conclusion, eDNA is a valuable tool for blue whale conservation because it enables scientists to detect blue whale presence, monitor migration route, identify feeding areas, and support effective conservation planning. Thank you so much.
My name is Radin Atika binti Abdurrahman. Now that we have seen how eDNA works, let's look at its advantages and limitations in biodiversity conservation. So first, eDNA offers several important advantages. It is non-invasive, which means scientists can monitor blue whales without capturing, tagging, or disturbing them. This is especially important because blue whales are endangered and spend most of their time underwater. Secondly, eDNA enables the early detection of rare and endangered species. Even if researchers don't see a blue whale directly, traces of DNA left behind in seawater can reveal its presence. In addition, eDNA helps scientists track biodiversity changes over time and understand how marine ecosystems are responding to environmental change. This information can then support ecosystem and habitat conservation planning, help authorities protect important marine areas. However, eDNA is not perfect. DNA in seawater can degrade quickly, making detection more challenging. There is also a risk of contamination which may affect the result. Furthermore, eDNA cannot accurately estimate population size, and sometimes it cannot determine the exact location of an organism because ocean currents can carry DNA away from its source. To conclude, eDNA is a powerful molecular tool for biodiversity conservation. It enables efficient monitoring of blue whale and other marine species while minimizing disturbance to wildlife. Most importantly, it provides valuable scientific data that helps researchers and policymakers make informed decisions to protect marine ecosystems and preserve biodiversity for future generations. That's all from us. Thank you.